Welcome to lecture 18, DNA Technology and Genomics. So today we are going to be looking at gene cloning, genetically modified organisms, also known as GMOs, DNA profiling, and the general idea of genomics. So let's start with gene cloning. So genes, which we learned about in earlier lectures, can be cloned in what's called recombinant plasmids. So here are some important terms that you're going to need to know. So biotechnology, which we often refer to as biotech, is the manipulation of organisms or their components to make useful products. Now, humans have actually been doing some form of this for thousands of years. We have been manipulating organisms or components of them, such as microbes, to make wine and cheese, and selectively breeding stock animals, dogs, and other animals. DNA technology is the set of modern laboratory techniques used to study and manipulate genetic material. Genetic engineering involves manipulating genes for practical purposes, and gene cloning leads to the production of multiple identical copies of a gene-carrying piece of DNA, what we call in vitro, which means in a test tube, to form a single DNA molecule. So we also have what's called recombinant DNA, and recombinant DNA is formed by joining nucleotide sequences from two different sources and sometimes from different species. So we're going to speak briefly about how this happens in bacteria, because we use bacteria quite a bit for biotechnology. So one source will contain a gene that is going to be cloned. And then another source is the gene carrier. So we're going to end up inserting the gene from the first organism into the gene from the second organism. And we call that gene carrier, that second organism, a vector. You will also need to know the word plasmids, specifically when we're talking about bacteria. Plasmids are small circular DNA molecules that are found in bacteria. But they replicate separately from the much larger bacterial chromosome. So bacteria quite often has one large chromosome that's made up of DNA, but bacteria will also sometimes have what's called a plasmid, that little circular DNA molecule that replicates separately from the rest of the DNA, and these are often used as vectors. So the following are the basic steps in gene cloning. Plasmid, that little circular DNA molecule, is isolated. And then DNA containing the gene of interest is isolated. Plasmid DNA is treated with what we call a restriction enzyme. The restriction enzyme is going to cut that DNA in one place, which is going to open up that circle. Then DNA with the target gene is treated with the same enzyme, and multiple fragments are produced. And then plasmid and target DNA are mixed, and they're associated with each other. So then the recombinant DNA molecules are produced when the enzyme that we call DNA ligase, used kind of like glue, it joins the plasmid and the target segments together. The recombinant plasmid containing the target gene is then taken up by the bacterial cell. The bacterial cell reproduces to form a clone, and a group of genetically identical cells descended from a single ancestral cell arise. And this process of gene cloning can be used to produce a variety of desirable products. So let's take a look at that in picture form. So if you take a look here on the left, we have E. coli, which is a type of bacteria. You can see the larger bacterial chromosome here on the left, and then the plasmid, that circular bacterial DNA here on the right. Notice that they are separate, which is important. So when we start our gene cloning, we're going to isolate this plasmid. Again, this is circular DNA. So we're going to isolate that. Then in the other organism here on the right, so we have our E. coli on the left, we have a cell from something else here on the right. Notice that there is DNA already in the cell here, so we're going to isolate that DNA here in the other cell. Then going back to our E. coli on the left, the plasmid is going to be cut with an enzyme. So we're going to use that restriction enzyme to make a cut, opening up the circle. Now for step 4, let's go back to this other cell. we just isolated the DNA in this cell. So now this cell's DNA is cut with the same enzyme that we used to cut the plasmid. But notice in this cell, it's going to end up with many different fragments since we're looking at a different type of DNA here. So then the plasmid from E. coli has been opened up in the last step. So notice now there's a little gap. So then we are going to take the gene of interest. So this is the gene that we're interested in cloning that's coming from this cell here on the right. We just cut it out with that restriction enzyme. Now we're going to insert it into the plasmid that's coming from the E. coli bacterium. Then we're going to join them together with DNA ligase. That's kind of like DNA glue. So it's going to glue this gene of interest into the plasmid. And so now we have this recombinant DNA plasmid that includes the gene of interest from the other organism. So now that we have our recombinant DNA plasmid that includes our gene of interest, we're going to insert this new recombinant plasmid into the E. coli bacterium from the first step. And then we're going to let it reproduce like normal. So now we end up with clones of that cell that we just inserted the plasmid back into. But now it's going to be reproducing with that gene of interest in all of those little clones that it's making. We're able to harvest those. So once the E. coli bacterium or whichever bacterium it is that we are using reproduces, we're able to harvest the proteins that are made out of this. And we can also then insert genes into other organisms, but we are able to harvest it and use those proteins the way that we want. So quite often when we're talking about the gene of interest, it might be the gene that is used to code for a certain type of protein that we want to make. And so we insert it into the DNA plasmid, making a recombinant DNA plasmid, put it back in the bacterium, let the bacterium reproduce as it does, and then we're able to harvest the protein that's made from the gene of interest that's now being cloned. So let's visualize the concept. Enzymes are used to cut and paste DNA. So we talked about how there's that restriction enzyme that's used to cut it. We have DNA ligase that will then glue it back together. So let's take a look at restriction enzymes. So restriction enzymes that are used to cut DNA are able to recognize a particular short DNA sequence, which we call a restriction site. And when the restriction enzyme recognizes that restriction site, it then will cut both strands of DNA at that precise point in that particular sequence, yielding pieces of DNA called restriction fragments. Once cut, so once we have those restriction fragments, the fragments of DNA can then be pasted together by the enzyme DNA ligase. So the two enzymes that you'll really need to know from this are restriction enzymes. You can think of them like the scissors. Restriction enzymes are going to be cutting the DNA at specific points. And then when we want to put them back together, you can think of DNA ligase like the glue. So DNA ligase is another type of enzyme that is going to be gluing all of the pieces together. So if you take a look at that, every restriction enzyme recognizes one specific nucleotide sequence. And again, that specific nucleotide sequence that the restriction enzyme is recognizing and is known as a restriction site. So if you take a look, remember, here's all of our DNA in the double helix. We're going to focus on this particular piece right here. So the whole blue ribbon is DNA. But we are specifically interested in this straight little bit, and that's just an illustration so you can see what's going on. But this is the restriction site here. So whichever restriction enzyme we're using, its restriction site are these letters right here. So this is its restriction site. So the restriction enzyme is going to recognize the specific sequence at this restriction site. And remember, the restriction enzyme is like scissors for DNA, and it is going to cut the DNA at that specific site. And it's always going to cut the DNA sequences at that restriction site in an identical manner. So if you're reusing the enzyme, because remember, enzymes do not get used up. They do their business and then move on and get to do it again. So every time this restriction enzyme is coming to a restriction site, it's going to be cutting the DNA in the same way. So we're going to be getting the same results each time, which is important when we're cloning since we're trying to get identical things. So then a piece of DNA from another source is cut by the same restriction enzyme. And this is important because we want to be able to glue the pieces back together and they have to be able to fit. So you have to have a gene of interest from one organism that will fit just right into that plasmid. So it has to be cut the same, which is why we use the same restriction enzyme. So notice here that when we cut the gene of interest, so that's colored red, so you can keep it separate in your mind from what we're doing up here. So remember, the blue is from one organism, the red is from another. So in this red organism, in this red cell, this one has the gene of interest. So this red gene right here is what we want to clone. And so we're interested in whatever this gene is. We're going to use the same restriction enzyme that we used on this blue DNA up here. And it's going to cut it in the same way, making kind of a perfect puzzle piece. Now notice that we have these two ends that stick out. These are called the sticky ends. You can kind of think of that like we're going to insert it into the plasmid. We're going to glue it back together. And so, you know, you can think of like this is where the glue goes and it's kind of sticky. So these sticky ends are what is going to be attaching. Because if you look up here at the top, the original sequence up here was GAATTC. Notice down here, once we've cut it, we get just the G, but then we get the AATTC from the red gene. So these are going to fit together just perfectly. So we've got the G that's going to attach to the sticky end of this gene of interest. And then take a look at the bottom of the blue here. So we originally had CTTAAG. We're going to be cutting it here at the G. And notice now it's going to fit perfectly down here. So we've got the CTTAA is going to attach to this G. And then we've got our CTTAA, which is going to attach to that G. So just like puzzle pieces, they're going to fit together perfectly. We're using the same restriction enzyme, so we're getting the same cut in both places, so that way they fit together perfectly like puzzle pieces. And these ends that are sticking out here and here are known as the sticky ends. Notice I've said that several times, so you might want to remember what sticky ends are. So these sticky ends are going to be pretty much what we use to stick the gene of interest into the plasmid DNA here in blue. So that's what we're going to do next. We're going to stick them together. So notice we're inserting this gene of interest. They're able to fit together like puzzle pieces because the restriction enzyme cut them the same way. So we end up with the same sequences here. Just in between them, we now have the gene of interest. So think of it like puzzle pieces. And we're going to be using that DNA ligase to glue them back together. So now we have a complete plasmid that we're able to put back into the bacterium in order to reproduce and clone. All right, so let's move on to nucleic acid probes. Nucleic acid probes are able to label specific DNA segments. So let's take a look at that. So a short, single-stranded molecule of labeled DNA is called a nucleic acid probe, and that can tag a desired nucleotide sequence. You can see my picture of the book in the background. We quite often refer to this as a DNA library. We're able to create DNA libraries from this because when we talk about tagging a desired nucleotide sequence, what that basically means is we have a sequence of DNA that we want to find, but there's a lot of DNA in each cell, and we can't just sit there and go through the whole thing. So we need some way to tag it. We need some way to find it and basically like raise a flag and say, it's right here. The DNA sequence you're looking for is right here. And so that's what nucleic acid probes do. They're radioactive in some way. So quite often we can make them glow, or have something where we can recognize where they are. But that's why it looks like it's glowing, because quite often they do make it glow. And so they have this radioactive nucleic acid probe, which is a single-stranded piece of DNA. And it has the complementary sequence to what we're looking for. So if you take a look down here, what we want is to find where TAGGCT is in that DNA. So we make a complementary radioactive nucleic acid probe. So complementary means it has the base pair to it. So we're looking for a T. So we're going to create our nucleic acid probe with an A, because A is going to go find T, right? Then A, so our nucleic acid probe has a T, right? So it's going to be the opposite, the pair of what we're actually looking for. Make it radioactive, we make it glow. So then we release it, and it goes and it sorts through the different pieces of DNA until it finds the complementary pair to it, and then it's all lit up. And so it basically goes and it's like, I found it, it's right here, here's what you're looking for. So we use those nucleic acid probes to find pieces of DNA or find genes that we're interested in isolating or studying. All right, so let's look at reverse transcriptase, which can help us make genes for cloning. So a researcher can focus on the genes expressed in a particular kind of cell by using its messenger RNA as the starting material for cloning. So in this process, the messenger RNA, and remember we usually refer to that as mRNA, from a specific cell type is used as a template. And then something called reverse transcriptase produces a DNA strand from that mRNA. DNA polymerase produces the second DNA strand, and the DNA that results from such a procedure is called complementary DNA. And so I've referred to complementary DNA in the past. It represents only the subset of genes that have been transcribed into mRNA in the starting cell. So we're able to use this. We start with an mRNA template that has the sequence that we're looking for, and then we're able to use that in order to get a subset of genes that we're interested in studying. So the advantage of cloning with cDNA, that complementary DNA, includes the ability to study genes responsible for specialized functions of a particular cell type and obtaining a gene sequence that are smaller in size, easier to handle, and do not have introns. Because if you think back several lectures, we talked about how we cut out those introns, those non-coding pieces of DNA, or the non-coding pieces before they leave the nucleus. And so it's easier for us to look at smaller pieces of DNA that already have the introns cut out. And that's what we're able to do when we use this cDNA. All right, so let's move on to genetically modified organisms, also known as GMOs. Many of you have probably at least heard of these in reference to our food. So recombinant cells and organisms can mass produce gene products. And recombinant cells, like we talked about in the last couple of parts, recombinant cells and recombinant organisms are constructed by DNA technologies, and they're often used to manufacture many useful products, chiefly proteins. So quite often we use bacteria for this. They're often the best organisms for manufacturing a protein because bacteria have those plasmids, those circular bits of DNA, and phages, which are available for use as gene cloning vectors. So it's fairly easy for us to, isolate a plasmid, break it open, insert a gene of interest, glue it back together, put it back in the bacteria, let the bacteria reproduce, and get our clones that way. So that's fairly simple for us to do. It can be done pretty quickly and fairly cheaply. We can engineer them to produce large amounts of a particular protein. Bacteria are pretty good at reproducing, and they often secrete that protein directly into their growth medium, directly where they grow, which makes it fairly easy for us to harvest the proteins that are being made. So bacteria are some of the cheapest, easiest, and fastest ways for us to manufacture proteins. But the problem with that is that bacterial proteins are not always the same as eukaryotic proteins, because remember that bacteria are prokaryotes, and so there's only a limited number of proteins that we're able to make for human products or products that humans are going to use from bacteria. So sometimes it is necessary for us to use eukaryotes for this. So one of the common eukaryotes that we will use are yeast cells, because yeast cells are eukaryotes. Yeast cells are fairly easy to grow. And we've been fairly familiar with yeast because we've been using yeast to make bread and beer for a really long time. And so yeast was kind of a logical choice for us because it's a eukaryote, so it's a little closer to humans, easy to grow, fairly familiar with it because humans are real good at making bread and beer. Yeast can also take up foreign DNA and integrate it into their genomes. And they're actually often better than bacteria at synthesizing and secreting eukaryotic proteins, which again, makes sense because they're eukaryotes and bacteria are prokaryotes. So it makes sense that yeast would be better at taking up and synthesizing eukaryotic proteins. So mammalian cells, though, sometimes do have to be used for specific glycoproteins, which are proteins with chains of sugar attached. Some examples include human erythroprotein, or called EPO, which stimulates the production of red blood cells, factor VIII to treat hemophilia, and tissue plasminogen activator, TPA, which is used to treat heart attacks and strokes. So if you notice, all of these are medical uses. There are some very specific medical uses, and since they're very specific medical uses for humans, and humans are mammals, to make these specific things, we do sometimes have to use mammalian cells. So we have some options. Depending on the bacteria that we want to be cloning, or depending on the proteins that we want to be cloning, sometimes we'll use bacterial cells, sometimes we'll use yeast cells, sometimes we do need to use mammalian cells. So you can see here, here are the different products. So things like human insulin, human growth hormone, different things like this. So these are the products that we're making. You can see here who it is made by. So insulin, we're actually able to make using E. coli, that bacteria. There are some more down here where we have to use mammalian cells, and then you can take a look at the use. So, you know, human insulin is actually able to be made using E. coli type of bacteria. It's used to treat diabetes. So you can see the list here, what it's used for. Notice some of our very specific medical uses. We are using the mammalian cells here at the bottom. So you can imagine this is used quite a bit by pharmaceutical researchers, and pharmaceutical researchers are currently exploring the mass production of gene products. We're pretty good at some of these. You know, we're pretty good at making insulin. We're pretty good at making human growth hormone, although we're always trying to get better at it so we can make them cheaper and more readily available to the general public. But pharmaceutical researchers are constantly studying not only how to improve upon technologies that we already have, but also for new ones, including some of the current research is looking at how we make whole animals or whole plants. And some of the other ones that you might be interested in or that you might have heard of are these pharmaceutical researchers or these scientists are trying to make edible meat that was never actually a living organism in a lab. So they've had a little bit of success with that. This is very difficult technology for the most part, but it is something that has the possibility of really changing human lives, of really improving the medical field. And so it's something that a lot of people are studying. And so it's been kind of slow going, but we've had some major breakthroughs. And you know, you can see with the current situation that when scientists are motivated and more importantly funded, that they can get quite a bit done, even in a short amount of time, if given the proper resources. So there is such a thing as recombinant animals. And so that is, like we were talking about on the last slide here, how they're trying to mass produce the genes of whole animals, or they're trying to make a whole animal. We call those recombinant animals. Recombinant animals, though, are really difficult to reproduce. You can imagine it's really hard to mess with the DNA and make a fully functioning live animal. It's also really, really expensive. It's used to clone to produce more animals with the same traits. So it could be really useful, you know, because if we want an animal with a specific trait, we would be able to isolate that gene, you know, insert it back in, and then just clone those animals in order to get the recombinant animals, those specific traits that we want. But again, it's very difficult, it's very costly, and there's also a lot of moral questions surrounding it. So it could be really useful, but as of right now, it's hard, it's expensive, and it's kind of morally questionable, depending on who you're talking to. So DNA technology, like we were just talking about, has changed the pharmaceutical industry and medicine. So DNA technology, including gene cloning, is widely used to produce medicines and to diagnose diseases. So there are therapeutic hormones that are produced by DNA technology. This includes insulin to treat diabetes, human growth hormone, and that tissue plasminogen activator, or TPA, which is a protein that helps dissolve blood clots and reduces the risk of subsequent heart attacks. DNA technology is used to test for inherited diseases. It's used to detect infectious agents such as HIV. It's used to produce vaccines, harmless variants or mutants or derivatives of a pathogen that stimulate the immune system to mount a lasting defense against that pathogen, thereby preventing disease. So DNA technology is really useful for medicine. So here's the connection. Genetically modified organisms are also transforming agriculture. So let's take a look at our food right now. So this isn't a new idea. Since ancient times, for thousands and thousands of years, people have been selectively breeding agricultural crops to make them more useful. One of the most common ones that you can think of is, instead of spraying with pesticides, we are trying to get crops that are just naturally immune to certain pests or just naturally fend off certain pests. We can also use them to make plants edible. Like if you've never seen wild types of the fruit that you eat, look it up, because most of the stuff that we eat has been selectively bred for a really long time to remove a lot of the seeds, to make the, it has a peel, to make the peel so we can actually peel it, to make the flesh easier for us to eat. So look up the wild types of the fruits that you eat and you will be astounded at how different these are. But we don't even think about it because this is a process that's been going on for thousands of years since people started agriculture. So this isn't a new idea that we're selectively breeding crops, but it is really coming to the forefront right now since the introduction of DNA technology. Because, for thousands of years, we've been selectively breeding. So we know that, this is a trait that we want. We want less seeds, right? We don't want to just be choking on all of these seeds. So every generation, we're going to find individual plants that have the least amount of seeds. We're going to breed those. And in the next generation, we should get a crop that has less seeds than the one before it. And out of that crop, we're going to find the individuals that have the least amount of seeds, and we're going to breed them. And so then in the next generation, we have even less seeds, and so on and so forth until we get a plant that's really edible for us. So that we've been doing for a long time. But now we have DNA technology, which is quickly replacing that traditional breeding program, because now instead of waiting generation after generation after generation, having to find those individuals, breed them, hope they reproduce, find the individuals in the next generation and then wait generations and generations, now we have this DNA technology where we can actually identify a gene, we can manipulate a gene that we want, and then clone that to get the output that we want. And so DNA technology is basically used as a faster way of those traditional breeding programs. And we're able to do that in both plants and animals. And you're seeing that a lot in current agriculture in order to get the plants that are the most edible, the plants that can resist pests, the plants that can withstand drought and other effects of climate change. And so there's a lot of potential of how we're going to use this and how we are currently using this. So genetically modified organisms, or GMOs, contain one or more genes that are introduced by these artificial means, and transgenic organisms contain at least one gene from another species. And so sometimes what we do is we take a gene from another species, like say there is a similar species that is drought tolerant or pest resistant. We can identify, we can isolate that gene, and then we can insert that into the other crop that we're interested in order to make that crop more drought tolerant or more resistant to pests or disease. And so those are known as transgenic organisms, when they have at least one gene from another species. So this idea of genetically modified organisms it's new in the sense that we are introducing the genes by artificial means, but the basic idea of genetically modifying organisms in general is not new. We've actually been doing that for thousands of years through selective breeding. But what's new about GMOs in particular is that we're doing this through artificial means. And so that's where, because you hear a lot of moral debates about GMOs and how GMOs are great, GMOs are bad for you, GMOs are not moral. That's where it's coming from. It's not really the idea of us genetically manipulating organisms, because we've been doing that for a really long time and people didn't have a problem with it. It's the idea that we're doing this by artificial means. but there are a lot of really good uses for it. You know, we are able to quickly produce food that is drought tolerant or tolerant to pests, or easier for us to eat, or grows faster. And so that is something that you can think about. So if you take a look here, here's an example. So here's our plasmid. Remember, that's that circular piece of DNA found in a bacteria. In this case, it's agrobacterium. Here's our restriction site that the restriction enzyme is going to recognize. It's going to be cut at that restriction site. Then we have the DNA containing the gene for the desired trait. It's going to be inserted into the plasmid, creating a recombinant plasmid, glued back together using DNA ligase. It's then inserted into a plant cell. So here's where it's a little bit different. We're now inserting it into a plant cell. So this desired trait could be something like drought tolerance, or resistance to pests, or resistance to some kind of disease. So we're then going to insert that recombinant plasmid into a plant cell. So now the plant cell is going to have that gene that codes for some type of drought tolerance or pest tolerance or something like that. And then the plant cell is going to reproduce and grow into a plant. And now we have a plant with that new trait. So it's fairly similar to what we were doing before, but now we're inserting it into a plant cell in order to get crops with the desired traits that we're looking for. So how do we connect this to humans again? Gene therapy may someday help treat a variety of diseases. So gene therapy is the alteration of a diseased individual's genes for therapeutic or medicinal purposes. So one possible procedure that we could do is a gene from a healthy person is cloned, is converted to the RNA version, and then inserted into the RNA genome of a harmless virus. Bone marrow cells are taken from the patient and infected with the recombinant virus. Now remember, infection is not always a bad thing. We think of it like, oh no, you've been infected with a virus. But in this case, we're taking a harmless virus, so it's not going to harm the host. Instead, we are going to have the RNA version of a healthy gene inserted in there. So in this case, infection is actually going to be a good thing. We want this virus to be infecting the patient because it's carrying a good gene. The virus inserts a DNA version of its genome, including the normal human gene, into the patient's cell's DNA. The engineered cells are then injected back into the patient. So we've got a healthy person here. We clone one of their genes, a normal allele. An RNA version of a healthy human gene is inserted into a retrovirus, that harmless virus. So here's the RNA genome of the virus. There's the retrovirus. Bone marrow cells are infected with the virus. The viral DNA carrying the human gene inserts into the cell's chromosome. This is the bone marrow from the patient. And then that engineered DNA, the engineered cells are injected back into the patient. And so now you have those healthy cells that are going to be spread throughout the patient. And the idea here is that now they have healthy cells, which should hopefully help them to, which might work as a cure. So the promise of gene therapy far exceeds actual results. This is all, you can kind of see me being like, well, and then we insert it here, and then we kind of do this, and then we put it back in, and then it kind of works, and it's kind of vague how we're doing this. We haven't had a ton of success with this. There has been some success, but this is really complicated. So, just remember that the idea here is that we're taking a gene from a healthy person, we're turning it into the RNA version, we're using bone marrow and a virus to try to insert that healthy gene into a patient who has the diseased gene. But this is really complicated. We've had a little bit of success with SCID and LCA, but for the most part, this is something that we are still really working on because it's really complicated to do. But if we can figure it out, it has so much potential to help people. So it's something that there are a lot of people working on. However, the use of gene therapy raises a lot of ethical questions. So some critics suggest that tampering with the human genome will invariably lead to the practice of eugenics, which is the idea that we are deliberately controlling the genetic makeup of human populations. If you've never seen the movie Gattaca, watch the movie Gattaca, because this is what it's about. It's basically looking at a futuristic society where the people in the higher socioeconomic classes are able to manipulate the genes of their offspring. And so when you find out that you're pregnant, you go in and they're like, what color would you like your kids' eyes to be? They program that in. What color would you like their hair to be? You know, how fast would you like them to run the mile? You know, and so they're able to edit the genes of these offspring while they're in utero. And so this is something that's obviously really expensive. So all the rich people do this and they make these like perfect little babies who then control society, and all the poor people can't afford it. And so they end up with these babies who are not gene edited. And so then there's this whole like underground movement, because only the, you know, the babies that have had their genes edited are able to get the good jobs. And so there's this whole like underground movement of how you can like, you know, get in and fake tests. And it's really interesting and asks a lot of really good ethical questions about eugenics. So Gattaca, look at the movie Gattaca if you've never seen it. But there are other observers that see no fundamental difference between the transplantation of genes into somatic cells and the transplantation of organs. You know, so we do organ transplants, right? People seem fine with that. So there's this idea of, could we then grow a human heart in a lab? You know, or how is gene editing really any different than that? So it's a really big ethical debate that you think about it. All right, so now let's take a look at DNA profiling. So we're still looking at DNA, but we're gonna shift a little bit. There's still a little bit of gene cloning going on here. We're gonna be cloning and sequencing parts of DNA. This is used a lot in criminology. And so if you're thinking of crime scenes and you know, trying to catch the bad guy, this is where you recognize this word from, this DNA profiling. So let's take a closer look. So the analysis of genetic markers can produce what's called a DNA profile, which is kind of like an identification card for someone based on their DNA. So DNA profiling is the analysis of DNA samples to determine whether they came from the same individual. So for those of you who are still thinking like, I have heard the term DNA profiling, you probably heard it on a show like CSI, or you know, some kind of like crime drama where they're trying to catch the bad guys and they find like a little piece of hair or a little speck of blood, and then they're able to run that and find out who the bad guy was. That's through a process called DNA profiling. So that's this next section that we're going to talk about, is how crime scene investigators and detectives are able to take a little piece of hair or a little speck of blood and use it to find the bad guy. So that's DNA profiling. So in a typical investigation, which involves DNA profiling, DNA samples are isolated from the crime scene, the suspects, the victims, or other evidence, because we might find a little piece of hair, we might find a little speck of blood at a crime scene, but we have to have something to compare it to. So either we need to have someone's DNA already on file, or we need to get a sample. So quite often, you know, you'll see in the crime dramas where they have, you know, a set of suspects and they go and like swab their cheek. They're getting a DNA sample from that person to compare with DNA they found at the crime scene. Selected markers from each DNA sample are amplified. That means they're copied many times. Quite often when we find a little piece of hair or a little speck of blood, it only has a really small amount of DNA. And in order to profile the DNA, we need to have a larger amount. And so we take that small piece of DNA and we copy it until it's big enough that we can actually analyze it. So we're gonna have a bunch of copies, but it's going to be big enough for us to at least look at what those copies are. The amplified DNA markers are then compared, providing data about which samples came from the same individual. Because if we have samples from the same individual, we should be able to see, oh, the DNA that came from the crime scene is the same as the DNA that came from, you know, suspect number 2. So that's the basic idea of DNA profiling. So DNA is isolated, right? We found a little drop of blood at a crime scene. There are 2 people that we think might have done it. And so we are going to collect DNA samples from suspect 1 and suspect 2. The DNA of selected markers is amplified. So we say we're going to look at this particular piece of DNA from both the crime scene, suspect 1 and suspect 2. We're going to amplify it. We're going to make more of it. And then we're going to compare it usually through a process called gel electrophoresis, which I'm going to talk about in a minute because that's the fun part, and that's the part that you're going to practice doing. So then we compare them. So if you take a look here, we're basically just looking for identical pieces. So if you take a look, you'll notice this is the DNA from the crime scene. This is the DNA from suspect 1. Notice it doesn't look the same, so it was not suspect 1. We take a look at suspect 2. The lines are in the same place, so these are lining up. That means that this DNA from the crime scene matches suspect number 2. Suspect number 2 is your bad guy. So, basic idea of DNA profiling. Now we talk about amplifying DNA, that quite often when you find a little drop of blood at a crime scene, it's not actually enough for us to profile. And so we have to amplify it. And we do that through a method called PCR, or polymerase chain reaction, which is basically just copying that small amount. It's a technique by which a specific segment of DNA can be targeted and then quickly amplified in the laboratory. So we want that little piece, but it's not big enough for us to actually profile. So we're going to take that little piece, we're going to use PCR to amplify it to make a bigger sample in the laboratory. So then we can actually analyze it. PCR, polymerase chain reaction, relies upon a pair of short primers, which are chemically synthesized, so we're making them, single-stranded DNA molecules with sequences that are complementary to sequences at each end of the target sequence. So we've talked about this a little bit before, about the complementary strands, so they're going in and looking for their base pairs and using that to identify the sequence that we're actually looking for. So one primer is complementary to one strand at one end of the target sequence, and then a second primer is complementary to the other strand at the other end of the sequence. The primers then bind to sequences that flank the target sequence, that are on either side of the target sequence, marking the start and the end points for the segment of DNA that's being amplified. So we send in these two primers, one attaches to one end, one attaches to the other, and then we know it's the stuff in between that we're actually looking to amplify using PCR. So the basic steps are PCR is that a reaction mixture is heated to separate the strands of DNA in the double helices. So remember, we have that double helix. We're going to heat it so the double helix kind of breaks apart into two separate strands of DNA. The strands are cooled down. As they cool, those primer molecules hydrogen bond to their target sequences. Remember, one on one side, one on the other. A heat-stable DNA polymerase builds new DNA strands by extending the primers in a 5-to-3 direction, just like when we talked about DNA sequencing or DNA replication. And then these 3 steps are repeated over and over again, doubling the amount of DNA after each 3-step cycle. So here's our target sequence, right? We're still in the double helix here at the beginning. We're going to heat the double helix so it separates. We are going to insert the primers that bond with the ends of the target sequence. DNA polymerase is going to work in the 5' to 3' direction in order to duplicate the DNA. Then we're going to keep doing it over and over again until we have enough DNA from that target sequence in order to send it on to the next step in DNA profiling. So we're still kind of at that second step. We have our samples. Now we need to amplify the target sequence enough so we can send it on to the last step. The advantages of PCR are that it can amplify DNA from a very small sample. It's fairly quickly. When I was an undergrad, we actually did PCR over the course of just one lab. And it's a reaction that's highly sensitive, copying only the target sequence. When we first started analyzing DNA from crime scenes, what was happening is that we were finding that we would get hair or blood or something with DNA in it, but there just wasn't enough DNA for us to actually run a profile on. And so it wasn't until the introduction of PCR that DNA profiling actually became something we could reliably do fairly quickly and easily in order to help us catch bad guys. So devised in 1985, PCR has had a major impact on biological research and biotechnology. PCR has been used to amplify DNA not only from crime scenes, so we don't just use this in crime scenes, but we also use PCR to amplify fragments of ancient DNA from mummified humans. We used it to amplify the DNA of a 40,000-year-old frozen woolly mammoth, a 30-million-year-old plant fossil, and even DNA from fingerprints or from tiny amounts of blood, tissue, semen, things found at crime scenes. So we talk about this a lot because it's really popular on shows like CSI. But if you think about other DNA that we might be interested in analyzing where we only have small fragments, we only have small amounts, would be things like from mummies or from frozen animals, from ancient things where we're just finding little pieces of them. They can look at a 40,000-year-old woolly mammoth and maybe there's just like a strand or two of hair left. There's not too much DNA there, but wouldn't it be interesting if we could somehow analyze the DNA that is there? PCR is what makes that happen. It's able to amplify the DNA enough that we can actually send it on to the next step, which is gel electrophoresis. Now, this is my favorite part of DNA profiling because this is something that we can actually do in the lab. So many DNA technology applications rely on gel electrophoresis, which is a method that separates macromolecules, usually proteins or nucleic acids, based on their size. So that's what's important, is we're going to be looking at a gel. It's called gel electrophoresis because you're literally putting samples of DNA into a gel. It goes on your desk. It's literally a piece of gel. When we're in person, you actually will mix and make the gel, and then electrophoresis, because we're using electricity to make the DNA move. And it's being separated by size. So that's going to be important, is that when we're doing gel electrophoresis, it's going to be separating DNA by size using an electrical charge, usually an electrical charge. So gel electrophoresis can be used to separate DNA molecules based on size. And here's what happens, is a DNA sample is placed at one end of a porous gel. So I mentioned we actually will make a gel. It's usually like yay big. We use this little comb to poke holes in one end, and then we put a sample of DNA in each of those wells, each of those holes that we just poked. And so we can put, a DNA sample from the crime scene in the first well, and then from suspect 1, and then suspect 2, and then suspect 3, and suspect 4 in each of the subsequent wells. And so we put that on one side, and then we put the lid on and we hook it up to a current. So we hook it up to electricity. And DNA molecules move from the negative electrode towards the positive electrode. So if we have the wells on this side, we're going to hook it up to a negative charge on that same side, and it's going to move towards the positive side. Shorter DNA fragments are going to move through the gel matrix more quickly. Think about if I were to tell you right now to go run as far as you can, and you go run as far as you can, then I give you time to recover, and I put on a 100-pound weight vest and tell you to go run as far as you can again, you're probably not going to run as far with a 100-pound weight vest on. So you can think of that kind of similar to the gel electrophoresis. The smaller DNA fragments are going to move more quickly, so they're going to move through the gel more quickly than the large fragments. And so we end up getting these bands in the gel, and the ones that don't move very far are the bigger pieces of DNA. The ones that move all the way across the gel are the smaller pieces of DNA. So again, we're separating them by size using an electrical current. The DNA fragments appear as bands, which I'll show you a picture of in just a minute. And we're able to visualize that. We're able to see them because we stain them using some kind of radioactivity or fluorescence. So they're different colors, basically. You'll see that in the picture in a minute. And so each band is a collection of DNA molecules of the same length. And so if we're looking at our gel again, everything in this band is going to be the same size. Everything in this band is the same size. So separating by size, smaller pieces move more quickly and will therefore move farther across the gel than the larger pieces. So let's take a look at that. So here's our gel. So you can imagine this is like an actual gel. It's not the same thing as Jell-O, but if you need to think of it like Jell-O, it's kind of that Jell-O consistency. So it's an actual gel. We make these wells on one side where we insert our DNA samples. So we can have DNA from the crime scene here in well number 1. This could be suspect number 1 and suspect number 2. We're then going to hook it up to an electrical current, positive on the same side as the DNA, or sorry, negative on the same side as the DNA, positive on the other side. We're going to turn that electricity on, and the DNA that's on the negative side is going to be attracted to the positive charge on the other side, and it's going to start to separate. And it's separating by size. Smaller, shorter pieces of DNA are going to move faster. So the ones that are down here at the bottom by the positive side, these are going to be the smaller pieces of DNA. The ones up here at the top that didn't move so far, they're slower. Those are the larger pieces. And we're going to end up with these bands. And so each of these bands represents a piece of DNA, and we've added something to it that makes it glow or be a certain color or something that we can see. When we're looking at it, looks like somebody like drew on it with marker kind of. So you can see here, sometimes they glow, sometimes they're just different colors. But then we're able to compare, if this is the DNA from the crime scene, does it match the DNA from suspect number 1 or the DNA from suspect number 2? You're just lining up these bands. So you take a look. There's a band here. Is there a band here? Is there a band here? And you match them up. You see if any of them match. So if you're looking at this one, you'll notice if this was the crime scene, suspect 1 and suspect 2, none of them match. So suspect 1 and suspect 2, probably not your people. So there's something called short tandem repeats that are used as a type of analysis in DNA profiling. So let's take a look at that. So repetitive DNA consists of nucleotide sequences that are present in multiple copies of the genome. Short tandem repeats, STRs, are short nucleotide sequences that are repeated in tandem. They're composed of different numbers of repeating units and individuals that are used for DNA profiling. STR analysis compares the lengths of STR sequences at specific sites in the genome, and it typically analyzes 13 sites scattered in the genome. So we usually keep it consistent. So you can take a look. Here's the crime scene DNA at the top. Here's the suspect's DNA at the bottom. So we've got an STR site number 1, we've got STR site number 2. We're going to be looking at the length. So here's site number 1. We're going to start here, and notice we're going to be counting how many of these short tandem repeats match. So in the case of STR site 1, notice that we have the same number at the crime scene as we do in the suspect's DNA. So these are a match. But if you take a look at site number 2, you'll notice here we've got 1, 2, 3, 4, 5, 6, 7, 8 from the crime scene. But if you take a look at the suspect's DNA, we've got 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13. Those don't match. So we have our amplified crime scene DNA in here, our amplified suspect's DNA. We can run gel electrophoresis. Again, the larger or longer fragments are here at the top, shorter, smaller are here at the bottom. And again, notice that while one of them matches, this orange one matches, the purple ones do not. So what we just saw here, notice how the purple, the suspect's DNA was larger than it was at the crime scene DNA. Same in the orange, different for the purple. Notice when we actually run that through gel electrophoresis, you can see that. So you can see that the orange was the same length because it's showing up at the same place in the gel. But if you take a look at this crime scene DNA for purple, you'll notice that it's smaller because it went farther. We know that it's smaller because it went farther. And then this piece from the suspect's DNA was larger because it didn't go as far. So we can see that these are not a match. So DNA profiling has provided evidence in many forensic investigations. It is commonly used to match suspects to crime scenes currently, but we've also used it retroactively to take a look at old crimes as well. It's been used to clear the name of a lot of people who have been in prison, falsely convicted. So DNA profiling is used to determine guilt or innocence in a crime. It's also used to settle questions of paternity. And it's used as to probe the origin of non-human materials. So I talked a little bit about the Innocence Project. I'll give it a name now. It's called the Innocence Project. And so when DNA profiling came out, there were lawyers who recognized that this had a big potential to clear the names of people who had been falsely convicted before DNA profiling was a thing. And so before we were able to use DNA in crime scene investigations and in trials, we were looking at evidence. And so there were a lot of people who were wrongly convicted. And then once the DNA technology came out, we had saved samples from the crime scene. And so we're able to go back and do DNA profiling on these old cases by getting a sample of DNA from the person who was already in prison and compare it to DNA that had been found at the crime scene and then just saved. And so there was actually a lot of people whose convictions were overturned when we were able to actually compare their DNA to the DNA found at the crime scene. So the Innocence Project is a nonprofit organization that was dedicated to overturning wrongful convictions using DNA technology and some legal work. And they've been able to exonerate more than 300 convicted criminals since 1989. So over 300 people who were wrongfully convicted were able to have their sentences overturned by the introduction of DNA technology. And there were 17 of those who were on death row, so for crimes that they didn't commit. We were able to show that using DNA profiling once that became a technology we could actually use. And it's relatively new. Look at that. So the Innocence Project was since 1989. There's also been some really creative ways that we've used this. So one of the strangest cases of DNA profiling is that of the Cheddar Man, who was a 9,000-year-old skeleton found in a cave near Cheddar, England. Scientists were able to extract DNA from his tooth and use DNA profiling to analyze it. And what they found was that the Cheddar Man was actually a direct ancestor through approximately 300 generations of a present-day school teacher who lived only half a mile from the cave. So it looks like his descendants didn't move too far. But that's an interesting example of how we've been able to use DNA profiling. All right, so our last section in this lecture is genomics. So let's take a quick look at genomics. So genomics is the scientific study of the whole genome. So trying to map the entire genome of a species. And the study of an organism's complete set of genes and their interactions. The initial studies for genomics focused on prokaryotic genomes. Many eukaryotic genomes have since been investigated. And as of 2013, the genomes of nearly 7,000 species have been completed, and there's thousands more in progress. So again, scientists are studying the entire genome, the entire set of DNA found in an organism, and they're trying to catalog it for all of these different species. So you can see some of the ones that have been done. You know, in 1995, we completed the genome for a certain type of bacteria. There were 1,700 genes there. We've done it for yeast, we've done it for nematodes, fruit flies, mice, rice, humans. Look, we did humans back in 2003. I actually did a high school science report on the Human Genome Project, which had not been completed when I did my report. Chimpanzees we've done, which is interesting. We're gonna talk about that in just a minute. Macaques, wallabies. But anyway, you can see that we started off kind of small. We started off with bacteria because they don't have as many genes. Then we kind of worked our way up to some larger organisms, some ones that are a little harder to study the genomes. But we got some that we're really interested in. Take a look at the number of genes in rice, people. Look at that, 42,000. Rice has a lot of genes. And now we're into some more complex ones. So it takes a lot of money and a lot of time. But we get some really interesting results when we're able to study the entire genome of a species. Genomics allows another way to examine evolutionary relationships. So genomic studies showed that there's a 96% similarity in DNA sequences between chimpanzees and humans. So chimpanzees and humans are both great apes, and their DNA is actually very, very similar. So we share 96% of our DNA with chimpanzees. Functions of human disease-causing genes have also been determined by comparing human genes to similar genes in yeast. So lots of different ways that these genomic studies can be used. The Human Genome Project revealed that most of the human genome does not consist of genes. So back when I was in high school, I took a forensics class, and I mentioned that this was back like before the Human Genome Project was completed. And so it was kind of a new technology that we were looking at. And back then, I remember the teacher teaching us about junk DNA, about how most of the human DNA is junk DNA and it doesn't really do anything. That's not entirely true. A lot of the human genome is non-coding, so it doesn't consist of genes, but that doesn't mean that it's junk. It doesn't mean that it's useless, which is something that we've just been learning fairly recently, that even though most of the human genome doesn't code for genes, it still serves a purpose. So the goals of the Human Genome Project, trying to examine the entire human genome, was done in order to determine the nucleotide sequence of all DNA in the human genome and identify the location and sequence of every human gene. Imagine if we do that, and we do now because we did finish this project. But the results of the Human Genome Project indicate that humans have about 21,000 genes in 3 billion nucleotide pairs. Only about 1.5% of our DNA codes for proteins, tRNAs, or rRNAs, and the remaining 98.5% of the DNA is non-coding. Doesn't mean it's useless though. So if you take a look, here are exons, here are our coding parts, again, only about 1.5%. And back when I was in high school, this pie chart looked something like, here's the useful part of DNA, here's the junk, non-useful part of DNA. You can see that's not true. The non-coding parts do stuff. So we have introns and regulatory sequences that account for about 24% of our DNA. We have unique non-coding DNA, accounts for about 15%. We have repetitive DNA, accounts for about 15%. That's unrelated to transposable elements, about 44% repetitive DNA that includes transposable elements and related sequences. One of the biggest advantages to having non-coding DNA is if there's a mutation in a non-coding part of DNA, it usually doesn't matter. And so by having only a small percentage, only 1.5% of our DNA actually code for proteins, code for genes, that's a very small percentage of DNA that really matters if it's mutated. So if we get a mutation in this 1.5%, that can be really, really bad. But if we get a mutation in the rest of this, it often doesn't matter. And so these do other things. We have our introns, regulatory sequences, it does other stuff. But if it gets a mutation, it doesn't always matter. So that's one of the biggest advantages of having a large portion of our DNA be non-coding. It makes it less likely that mutations are going to affect what we're doing. So much of the DNA between genes consists of repetitive DNA. And so that's an important point too, is this 1.5% is not all bunched together. It's not like there's 1.5% of your DNA that codes, and then there's, the 98.5% that doesn't. They're kind of mixed up. So you'll have like a gene here, you'll have some non-coding parts, then you'll have another gene and some non-coding parts. So they're not all bunched up together. And much of the DNA between genes does consist of repetitive DNA, which are nucleotide sequences present in many copies in the genome. So a lot of times you'll just get a gene and then repetitive DNA, and then another gene and then repetitive DNA. Stretches of DNA with thousands of short repetitions are also prominent at centromeres and at the end of chromosomes, which we call telomeres. In a second main type of repetitive DNA, each repeated unit is hundreds of nucleotides long, and the copies are scattered around the genome. Most of these sequences seem to be associated with transposomal elements. We also call them jumping genes, which are DNA segments that can move or be copied from one location to another in a chromosome, and sometimes even between chromosomes. So genomes hold clues to human evolution. I mentioned earlier that humans and chimpanzees share a lot of their genome. They actually share about 96% of our genomes. About 1.2% in single base substitutions and about 2.7% in insertions and deletions of larger DNA sequences are the difference. So 96% is pretty much identical between humans and chimps. The remaining 4% differs 1.2% in single base substitutions and about 2.7% in insertions and deletions. So very, very similar, actually. Genes showing rapid evolution in humans include genes for defense against malaria and tuberculosis. So evolution quite often takes thousands or millions of years. But we have seen rapid evolution in things like defense against diseases, a gene regulating brain size, and what's called the FOXP2 gene, which is involved with speech and vocalization. So our vocalization, our speech actually evolved fairly rapidly. Neanderthals, which are a different species than humans, so they were a different species before Homo sapiens evolved, first appeared at least 300,000 years ago, and they were our closest relatives. They were a separate species from us, but they also had the FOXP2 gene, and they may have had pale skin, red hair, and we think they were lactose intolerant, which we can tell by looking at their DNA. All right, so now you should be able to explain how plasmids are used in gene cloning, explain how restriction enzymes are used to cut and paste DNA into plasmids. Remember, restriction enzymes are used to cut, get our DNA ligase, another enzyme used to paste. Explain how a nucleic acid probe can be used to identify a specific gene. Explain how reverse transcriptase is used to help make genes for cloning. We skipped over CRISPR, so you can read more about that in your book, but I'm not as concerned about that. Explain how different organisms are used to mass produce proteins of human interest. Explain how DNA technology has helped to treat, diagnose, and prevent disease. Explain what GMOs are and how they're transforming agriculture. Describe some of the risks posed to the creation and culturing of GMOs. Describe the benefits and risks of gene therapy and some of the ethical issues that arise, the basic steps of DNA profiling, including PCR and gel electrophoresis. Be able to read one of the gels to see if samples of DNA match each other, specifically as it applies to crime scenes. Explain short tandem repeats, the diverse applications of DNA profiling, think Cheddar Man. Explain how small segments of DNA can be sequenced directly and quickly. Describe some of the importance of the non-coding sections of the human genome. We did not talk about the whole genome shotgun method in the lecture, so you're welcome to read about that in your book. We also did not talk about proteomics, so you're welcome to read about that as well. And then describe the significance of genomics to the study of evolutionary relationships and our understanding of the special characteristics of humans. So that's the end of this lecture, and I will see you next time.